Today we will be talking about the gene expression process. Scientists say gene expression is like making cookies. The recipe is the genes, the ingredients are the amino acids, the dough is like the protein, and the oven is like the environment. The process of gene expression has three main stages: transcription, splicing, and translation. First step of gene expression. Transcription is a process in which information is rewritten. In biology, this process is basically the copying of a gene's DNA sequence and turning it into an RNA molecule. A segment of the DNA helix unwinds and unzips, and complementary RNA nucleotides from an RNA nucleotide pool in the nucleus pair with the DNA nucleotides of one strand. The RNA nucleotides are joined by an enzyme called RNA polymerase, and an RNA molecule results. The process happens in three stages: initiation, elongation, and termination. The first stage is initiation. The multiple subunits that make RNA polymerase assemble at the initiation site. During this process, RNA polymerase binds to a sequence of DNA called the promoter, which is near the beginning of a gene. Working along the DNA chain, the enzymes read the nitrogenous bases. Later, the DNA strands are separated by the RNA polymerase, providing the single-stranded template needed for transcription. The second stage is elongation. During elongation, the template strand, one strand of DNA, acts as a template for RNA polymerase. A chain that grows from 5 to 3 is created as the polymerase builds an RNA molecule out of the complementary nucleotides. Complementary or base pairing allows the two DNA molecules to be identical to each other as they have the same base sequence. The RNA transcript contains the same information as the non-template strand of DNA, but it contains the base uracil instead of thymine. Lastly, termination. Sequences called terminators signal that the RNA transcript is complete. At the termination site, the newly formed mRNA strand detaches and the RNA polymerase complex disassembles. Now, mRNA has a sequence of codons that are complementary to the DNA triplet code. However, transcriptions do not always turn out 100% correct. Mutations, such as substitution and elimination of nucleotides, might occur which can alter the function of the protein, which can lead to disorders. Splicing. Splicing is the editing of the messenger RNA, pre-mRNA, transcript into the mature messenger RNA, or mRNA. Splicing takes place within the nucleus either during or immediately after transcription. The key player of this process are the SNRPs, small nuclear ribonucleoproteins, or spliceosomes. The spliceosomes edit the mRNA through the following process. The spliceosome first removes the junk segments, introns, by pinching and cutting them out. They then attach the two neighboring exons, which then make up the mature mRNA and are therefore expressed. Translation. In the stage of translation, the mRNA is decoded to build a protein that contains a specific series of amino acids. After the mRNA strand has left the nucleolus and has been edited, it heads for translation across the membrane of the rough endoplasmic reticulum. Here it becomes associated with the ribosome, which is made up of two subunits, the large subunit and the small subunit, made of ribosomal RNA, rRNA, and proteins. The green spot marks the catalytic site, which catalyzes the reaction that links amino acids to make a protein. Translation proceeds in three phases. First, initiation. The ribosome assembles around the target mRNA. When the mRNA comes near these two ribosomal subunits, the two subunits sandwich the mRNA at the start codon. The ribosome reads the start codon, first three bases, then small molecules in the cytoplasm called transfer RNAs, tRNA, bring amino acids to the ribosome during elongation. mRNA codons are matched through complementary base pairing to specific tRNA anticodons. The ribosome matches the codon to the anticodon. tRNAs will bounce in and out of the ribosome at the A site until one has an anticodon that matches the mRNA codon. The ribosome has three sites for tRNA to bind. They move in the order of the amino acyl site, A, the peptidal site, P, and the exit site, E. Termination occurs when a stop codon is reached, and the ribosome releases the polypeptide to go do its job in the cell.